KCET and PU2 champions. I hope all my dear Chutku people are doing really good. So today starts the Masterstroke series for biology, and the first chapter that I have taken is Sexual Reproduction in Flowering Plants. We'll go chapter by chapter and we'll conquer everything. And I am pretty sure everyone has one doubt in your head. Ma'am, do you think this is enough for us to understand the concepts? If at all we are someone who do not know the basics of things, and you might have so many doubts around it, right? So the only simple answer that I want to give you is yes, it is more than enough. You do not need to watch any long lectures out there. This video would be enough. So please listen to me very carefully. Over here, I'll make sure all your concepts are super strong. I'll tell you what's more important for your PU2, what's important for your KCET. I'll show you a few questions for your KCET as well. And then we will be having a separate live lecture on question solving for KCET PYQs. Okay, I hope that's understood. So with that, Josh, do not go anywhere for the next 1 hour while I'll be teaching you. Here it's a teamwork, okay? You and me, we are going to do a teamwork, and I'll be giving you some homeworks to do, some small questions. Solve them up and put it down in the comment section. I hope you love the video, and my dear children, you can do it. You will definitely do it. Let's begin up with this chapter. So the first thing that we have is all about androecium. That is, sexual reproduction in flowering plants needs the male part as well as the female part, right? Then only sexual reproduction is going to happen. So the male part is known as androecium. And the easiest name is male reproductive system. Yeah, so androecium is a scientific name for the male reproductive part, and this androecium, guy, is made up of two things. Well, you know what? Androecium has also one more name, and that is stamen. So you can remember men as in male. So it is a male reproductive part. So this stamen or androecium is made up of two very important things. As you can see over here, this is your anther, and the next one which you have is what? Your filament. Ma'am, do you think this particular thing is important? Do we need to talk too much about it? Yes, because you got a KCET question based on this. You won't believe me, but listen to me carefully. So over here it is written, the stamen is divided into two parts. Agreed. Okay, there is a long slender thing that is known as what? Filament. This long slender thing is known as filament, and basically at the tip you have a bilobed anther, as in you can clearly see it has what? Two lobes. One lobe, second lobe. So basically it is a bilobed anther out there. Now, the filament's proximal end is attached to the thalamus or a petal. This is where from the question came from. So, the filament's proximal end, what do you mean by that? As in the closest end. So, this is the proximal end of the filament, and this will be the distal end. Distal is at distance. Okay, this is a distal end which you have, and this is going to be the proximal end which you have. The proximal end of it can be attached, can be on a thalamus, a structure that looks like this, or probably can directly be attached onto a petal as well. So question came that the proximal end of thalamus is attached to what? Anther, or basically thalamus, or basically petal, or you had options like that. Okay, so this point is super important for you to remember. So proximal here, distal here. Fine. Done. So this is the common structure of stamen that we have. Now we go deep dive into the structure of anther. This guy is super important. So anther, my dear children, if I zoom it in, it looks like this. So as I said, it is made up of two lobes. So it is a bilobed structure. Okay. Now within each lobe, you have some other thing. What is that other thing? It is known as theca. So basically, if I see this particular lobe, and if I zoom it in, all my dear children, come up with your binoculars. Here you can see a small ridge. Now I am not sure if at all you are able to see this, or on this side you can see a small ridge. Basically this ridge which is there is dividing the lobe into two parts. One part, second part. One part, second part. What do you call these parts as? You are going to call these parts as theca. What do you call this? Theca. So basically, for those of you who are not able to see this, I am going to make it something like this, right? Now, this is one part, this is second part, this is one part, and this is going to be the second part. What do you call this as? Theca. So one lobe has how many theca? It has two theca. One theca, second theca. So can I call that this is what? Dithecous condition. Di means two. Yeah, dithecous condition means two theca will be there. Cool. Fine. So lobes are there, it is bilobed, and there is a longitudinal groove. Longitudinal as in a line like that. It's a groove that is there which is dividing these lobes into what? Theca. Dithecous condition. Got it? Done, ma'am. Now, the anther is four-sided tetragonal structure consisting of 4 microsporangia located at the corners, 2 in each lobe. Ma'am, what are you saying? Listen, it's very simple. Over here, if I cut this thing off, okay, and I'm seeing from the top, how does it look? It looks like this. It looks like this. So over here, when I look at from the top, it looks like this. And why do I say that? So my dear children, this is going to be one lobe. This is going to be the second lobe. And each lobe is divided into 2 theca, right? So 1, 2, 3, 4. Dithecous condition is there. 1, 2 over here, 1, 2 over here, right? Now I am saying that basically it is tetragonal. It has 4 sides, isn't it? 1, 2, 3, 4. Yes, it is tetragonal structure. And each side will be having something like this, which you call as what? Microsporangia. One microsporangia will be there at each corner out there. This is microsporangia, or you can basically call it pollen sac also. So later on, this is something that is going to give us lots of pollen grains, which is why I can call it as pollen sac also. So we'll come back. So anther is 4-sided, got it. Consisting of 4 microsporangia. How many? 4 microsporangia located at the corners, 2 in each lobe. So in one lobe, you have 1 and 2. So 2 in each lobe will be there. Okay, done. Now, the microsporangia develop further and become what? The pollen sacs. They are going to develop further and they will be becoming the pollen sacs. So that is also clear. So anther is bilobed, it is dithecous, each lobe is dithecous, and not just that, in each theca you will be having 1 microsporangia each. Microsporangium each. And this microsporangium is what? The future pollen sac out there. Done? So this is also done. Now, the structure of microsporangium and the walls. So listen to me. When I say that you have microsporangium and all those things, you need to know the structure of it. Now this particular picture is super important for you. They can ask you the order in which the walls are placed. They can ask you about what wall is there for nourishment, which wall is there for protection, and all of it. So we should know their function also. So listen to me carefully when I say this. Over here, if this was your anther wall, it looked like this exactly. Right? Over here, the outermost wall that you have, it is known as epidermis. Cool? Epidermis. Now, inside that you have the next wall, which is known as endothecium. Okay? Inside that, rather than making what is there inside that, I am directly going to make something right over here. You know what is this? This is known as the sporogenous tissue. Sporogenous tissue. This is where the pollen grains are going to be formed, because this tissue is made up of so many cells. Tissues are made up of cells, right? And what do you call these cells as? Basically, you call the cells as microspore mother cell, MMC, or you can also call it pollen mother cell, because my dear children, they are the ones which are going to divide further and going to give rise to pollen grains, which is why I call this whole structure as what? Pollen sac. The microsporangium is basically what? The future pollen sac out there. So you have sporogenous tissue, and this is where you got to understand that the pollen grains are going to be formed. Now there should be someone to nourish this growing pollen grains out there, right? And what is that something out there? It is known as the tapetum. This layer that you see is known as the tapetum. And this is the 4th layer. Ma'am, how 4th layer? You said this is the 1st layer, this is the 2nd layer. Where is the 3rd one? 3rd one is not there. And then you said now it is the 4th layer. What is the 3rd layer? 3rd layer, Baba, comes in the middle of these 3 layers. It comes in the middle. And what do you call this layer as? It's very simple name. Middle layer. Okay? It is what? Middle layer. So I always tell you guys to remember it like this. When you go to doctors who check for ear, nose, throat, what do you call them? You call them ENT specialist. Okay. Similarly, over here, you have EMT. They are EMT specialist. Remember their order. It is super important. Now, these 3 of them, epidermis, endothecium and middle layer, these 3 of them are responsible for 2 things. One is to protect them. One is to protect the entire structure of the anther. Apart from that, they are also responsible for dehiscence of anther. What do you mean by that? Dehiscence means breakdown of anther. When these guys break down, that's when the pollen grains which are being formed over here, they are going to be released. So I have two functions to remember. One is what? Protection. Other one is dehiscence of anther. Only these three less. Okay, E, M, only these three less. What about T then, ma'am? What does T do? T is just chumma sitting out there? No, Baba. They are not chumma sitting out there. They are going to nourish the pollen grains out there. So your tapetum which is there, it is going to nourish the pollen grains out there, which is why it is unique in nature. For nourishment, obviously, you need to have a dense cytoplasm. What do you need to have? You need to have a dense cytoplasm because this cytoplasm could provide nourishment. Apart from that, you should also know one more feature of tapetal cells, and that is what? They are binucleate in nature. Binucleate means, bi means what? Two. Two nucleus will be there. So if at all I talk about one cell, they will be having two nucleus. Ma'am, why is this happening? So usually what happens? First of all, karyokinesis happens. That is the nuclear division. That is known as karyokinesis. After karyokinesis, there is cytokinesis that happens, which divides the cell into two. But here, karyokinesis took place, but cytokinesis didn't take place, which is why you have two nucleus out there in one cell. This is known as binucleate condition. And apart from being binucleate, they also have dense cytoplasm. Why do they have dense cytoplasm? Because they are the one used for nourishment of the pollen grains. Done. So these are the wall layers that you need to remember. E, M, T. Okay? Cool. Now, when I come back over here, I can read it out for you. So epidermis, endothelium, middle layer, tapetum, sporogenous tissue. For PUC champions, please understand that, where is it? You need to make the diagram of it. You would need to make the diagram of these wall layers. This is a very important picture which is given in your NCERT. So outer three layers is for protection and for anther dehiscence. I already spoke about that. Tapetum is there for nourishing the developing pollen grains. Remember that. And now tapetal cells have dense cytoplasm and often they are binucleate in condition. We know that. And the sporogenous tissue is the center. It later forms the pollen grains later on. Yeah, ma'am, we all know all this. Yes, you guys know all this thing, which is why I am telling you, when we learn something, we are going to learn it very nice in a way that we do not forget it. Now, I just want one thing from you before I even go ahead. That is, please let me know in the comment section, are you liking this video? And if at all you are, do not forget to hit the subscribe button right now. And do not forget to share it with your friends also who have taken biology. Yes, so subscribe to the channel, and if you've already subscribed, click on the like button, because when I come back and I see that, okay, ma'am, it helped, I get really happy seeing all this thing. Okay, and also, if you have any doubts which you have encountered until now, put them down also in the comment section below. Works? Yes. So let's go ahead. Let's see all about microsporogenesis. What do you mean by that? We spoke about microsporangium. Inside microsporangium, we told that basically there is sporogenous tissue which is going to form, which is having microspore mother cell or pollen mother cell, right? What are they going to form? They're going to form the pollen grains. And this process of formation, of breakdown of the microspore mother cell to later give down to microspores and later give down to pollen grains is known as microsporogenesis. Micro is for males. Okay, micro is for males, mega is for females. Okay, mega is for females, micro is for males, do not forget it. Cool. So let's see. So I told you there is microspore mother cell. There are so many microspore mother cell in what? In your sporogenous tissue out there. Out of them, when I talk about just one, all of them are dividing, I'm just talking about one, okay? So one microspore mother cell is going to start undergoing meiotic division. And you know the thing about meiotic division? At the end of the meiotic division, suppose I started with a cell which is having, suppose it is having 32 number of chromosome. Suppose it is having 32 number of chromosome. At the end, I will be having half of it only. So half of 32 is what? 16 number of chromosome. Why? Because we are making gametes. And gametes should be having half the number of chromosomes. Because suppose the male will carry 16, the female will also carry 16. This 16 and 16 when it comes together, it becomes 32 again. That is why we say that the gamete formation always require meiotic division. And meiosis, my dear children, happens in two stages. Meiosis requires two stages. stages. So first stage which is there, it is the reductional division which is going to happen. So suppose I started with a diploid cell out there. Diploid means 2n. Now it will become haploid over here. Haploid means half of it. So half of 2n is equals to what, my dear children? 2 get cancelled. So basically it will be just n. So half of it will be n. You call this condition as what? Haploid condition. So here we started off with diploid, and now it is going to be haploid in nature. This is meiosis 1, and I told you meiosis happens in what? 2 stages. So now again the cell is going to divide, one cell is going to divide into 2, one cell is going to divide into 2. Ma'am, again will it get halved? No, Baba. Only once it gets halved, and that is done. So here it will be the same number of chromosomes that we have. They are all haploid in nature. Baba, what do you call these cells as? You call them as microspores. But you know what? They are not separate when they are formed. They are all together. They are clumped up together, and which you call as microspore tetrads. You will call them as what? Microspore tetrads. Tetrad means 4 of them. Now these 4 of them are going to separate when there is going to be dehydration and everything out there. They are going to separate from each other. So when they are formed, they come in the form of microspore tetrads. Later this microspore tetrads, my dear children, later this microspore tetrads will be giving down to microspores. They will form microspores, and later these microspores will be forming what? Pollen grains. This is how things are going to go. Okay, so these microspore itself later forms the pollen grains. This is how things are going to go. So first of all, we have sporogenous tissue. Each cell, remember, each cell can form what? Microspore tetrad. Now microsporogenesis, meiosis in the pollen mother cell, or you also call this as what? You call this pollen mother cell because at the end anyway you get the pollen grains. You also call this microspore mother cell because before that you get the microspores, right? That is the reason. Now microspore tetrad initially grouped in 4, but later they separate. Maturation. Anther dehydrates, microspores dissociate and develop into what? The pollen grains out there. Pollen release is going to happen. Thousands of pollen grains are released when anther dehydrates. When anther gets broken down, thousands of them are going to dehydrate, as in they are going to come out as well. Done? Yes. Now we are going to talk about the pollen grain out there. So before I go ahead, everyone who is watching this video and who is preparing for KCET examination, my dear children, we have launched Abhyas KCET test series. It's strictly online. You can take it up. You can make the best use of it until your examination is done. So please do not be worried. There are 35 KCET mock tests. There will be chapter-wise KCET questions, and this is what I want you guys to attempt as well. So basically what you have to do is, once you're done with the Masterstroke, okay, this particular video, you know the concept. Now what do? Go attempt the questions. Go attempt the KCET PYQs out there, chapter-wise PYQs. It is definitely going to help you out. You do not need to solve all of them as of now, because I know you are busy with your PU2 syllabus preparation. So at least solve a few of them. Okay, at least solve a few of them, and all of it will be under timed condition. So 60 minutes, 60 questions, that's all. Okay, and the pricing for this is rupees 1499. And my dear children, this is one of the most trusted test series out there. We had multiple rank producers out there. For example, Prajwal with rank 171. Imagine out of 3 lakh children, if someone is getting 171, that's crazy, right? Kiran with rank 443, and the list goes on. Now those of you who feel that, ma'am, I do not want an online test series, I want an offline, we have officially launched Lakshya offline crash course as well. Not a series, it's a crash course. If you want to know further updates of it, put it down in the comment section, and always, and do not remember, do not forget, what am I saying? Do not forget, we will be coming up with a video on that as well really soon. Okay, fine. So go check out Abhyas KCET test series for sure. Suggest it with to your friends also. Okay, now going ahead, let's talk about the male gametophyte. What do you mean by that? Until now we formed the pollen grain. Where does this male gametophyte came from, ma'am? So this pollen grain which you're talking about, Baba, is the male gametophyte. Ma'am, why do you call this as the male gametophyte? Because it is the one. Pollen grain is not the gamete. Pollen grain has the gamete. Okay, pollen grain is something that is going to form the gamete. It has the gamete, which is why I call this as what? Male gametophyte. So PU2 children, the question might be coming, draw the structure of male gametophyte, or tell the features of male gametophyte. That means you are being asked about what? Pollen grains. You need to understand that. So now when you talk about pollen grains, the shape, the size of it is really important. The size is what? 25 to 50 micrometer, or you call this as nu meter. not micrometer, new meter in diameter. So 25 to 50 new meter in diameter is their size, and they are spherical in structure. Okay, now when you talk about how do they look and everything out there, we need to make a diagram. So over here I have already got your diagram, but as a teacher I am not really fine with what you call teaching like that. So let's make a diagram from the scratch. So over here you have a cell. Okay, this cell has a plasma membrane. This is the plasma membrane. You know, this is a very big cell. Over here, this cell has an irregularly shaped nucleus. This is also important for PU2 children. Irregularly shaped nucleus is out there. What do you call this cell as? This very big cell is known as vegetative cell. Vegetative cell. Very big cell. You can remember VVV like that. Now, inside the cytoplasm of this very big cell, vegetative cell, floats another small cell out there, which you call as what? Generative cell. Generative cell. And you know what? The shape of the nucleus is elongated. They have an elongated nucleus. So here irregular, here elongated. Now this generative cell, why do you think it is named like that? Because it is going to generate something. Generate what? What is missing over here? Male gametophyte should be having gametes, right? So this generative cell which is out there, my dear children, it is going to give down gametes. So G for generative and G for gametes, you can remember like that. How many gametes, ma'am? 2 gametes. So always remember, in one pollen grain, you have what? 2 gametes. You will be having 2 gametes. That is why it is known as gametophyte. So first of all, mitosis happened. Mitosis, when it happened, vegetative cell and a generative cell is being formed. The generative cell floats in the cytoplasm of the vegetative cell. Always remember that. Then this generative cell again undergoes mitosis. There is no meiosis. Meiosis happened and that's all. While microsporogenesis, meiosis happened and that's all. Okay, now we have formed the gametes with the help of mitosis. Done. Now this pollen grain which is there, it is covered by a layer which is continuous in nature. That means there are no breaks in between. This continuous layer is known as intine. Intine. And intine, my dear children, is made up of Priyanka Chopra. What do you mean by that? It is basically pectin plus cellulose. So PC as in Priyanka Chopra, or PC, you can remember computer PC, whatever you want to say, in whatever way you want to remember, you can. But this is intine, and intine is continuous layer made up of pectin as well as cellulose. Done. This is a very important concept for KCET children and also for everyone who is going to attempt your PU2. Cool. Now there is one more layer, and this layer is celebrated so much. Okay, this has huge fan base. Why? Because this layer, my dear children, is made of the toughest material out there on earth. And what is that toughest material out there? It is known as, first of all, what do you call this layer? You call this layer as exine. Okay, it is known as exine, and the toughest material with which it is formed is known as sporopollenin. Sporopollenin. Ma'am, why it is known as toughest material? Because if you put alkali, if you put acid, still it will just stay like that. It is just going to stay like that. There are not any trouble that is going to happen in the exine layer out there, which is why the pollen grains can be stored as fossils also. Why? Because it is made up of this sporopollen that you see right over here. Got it? This is super important. And also one very important thing, and that is, it is going to be a discontinuous layer. There is nothing over here. It is strictly discontinuous. Why it is discontinuous? Wherever the sporopollen is not there, there you have a gap. And this gap is known as germ pore. It is known as germ pore. So what exactly is going to happen with all this structure? Listen to me carefully. This is a pollen grain, and pollen grain will be landing on the female part, female reproductive part. The female reproductive part is made up of stigma, there is a style, and there is an ovary. Inside the ovary, you will have ovule, which will have the female gamete. So female gamete, for now, just understand, female gamete is right over here. The pollen grain which are there, they are going to come land on the stigma. And if everything goes well, they are going to form a tube-like structure. This is known as pollen tube. What do you call this? Pollen tube. Why is there a tube? Because with the help of this tube, the male gametes which are there in the pollen grain will be transported towards this area where you have the female gamete. Right? Now, who is responsible for formation of this tube, pollen tube, and everything out there? So, I'm going to talk about that also. Listen to me carefully. So, this intine layer that you see, the inner layer that you see, it is going to protrude outside. it is going to protrude outside. And inside that you have cytoplasm. Cytoplasm will be of what? The vegetative cell. Cytoplasm of the pollen tube will be of the vegetative cell, or basically the basic part of the pollen tube will be made up of the vegetative cell, which is why I will call this vegetative cell as tube cell also. I will also call this as tube cell. Why? Because it is going to help me with the formation of pollen tube. But always remember, it's the intine which is going to help in the border part. Inner things, yes. Inner content is from the vegetative cell, but who makes it? It is going to be the intine that is going to give the border. And they are going to move out through the germ pore. They move out through the germ pore. I hope this is clear. This is the only thing that you need to remember. I am going to repeat once more. Pollen grain has two cells, vegetative cell and generative cell. Vegetative cell has irregular nucleus and it is also known as what? Tube cell, because it is going to help with the content of pollen tube out there. Now the generative cell undergoes mitosis and it forms gametes. How many gametes? 2 gametes, my dear children. Okay. And now if you talk about plasma membrane, apart from plasma membrane, you have intine layer, which is made up of Priyanka Chopra, that is pectin as well as cellulose. That is continuous in nature. Exine is discontinuous because in some places you do not have sporopollen, and these places are known as what? Germ pore. And sporopollen is the toughest resistant material out there. You put alkali, acid, anything, it will be staying like that, which is why you can store pollen grains as fossils also. The diameter of this particular pollen grain is 25 to 50 nu meter. What? It is 25 to 50 nu meter. Got it? Now, one more very important thing. These are obviously released in flowering plants because flowering plants will only have this thing, right? You call these flowering plants as what? Angiosperms. What do you call them? Angiosperms. Now, there are two types of angiosperms. Okay, 60% of them are going to shed the pollen grain, are going to shed off the pollen grain in 2 cell stage. What do you mean by 2 cell stage? One vegetative and one generative. This generative is not going to divide now. While shedding off, they're just like that, they're just generative. While they reach over here, they will be doing mitosis and making the gametes. So 60%, most of them out of 100, 60% of them are going to shed off the pollen grains in 2-celled stage. This is also important. And rest, what do you have? 40% only. 40% of them are going to shed in 3-cell stage. How can it be 3-cell stage? One gamete, second gamete. So 3-cell stage. So 60% 2-celled and rest 40% will be in 3-cell stage. Got it? Yes, ma'am. Got it. And that's what about pollen grain as well. So over here I have explained everything. This is how it actually looks. I can show you a picture. So this is your tube cell or basically vegetative cell, and over here this is the small generative cell that you have. It has its own nucleus as well. Okay, the nucleus is elongated in shape. Here it is not been mentioned, but please remember it is elongated in shape. Okay, generative cell, if at all you talk about its shape, it is spindle shaped, but nucleus is elongated. Done. Okay, so now there is a question that we have to try together. Which of these options is true in the context of the above diagram of pollen grain? So this is a pollen grain where A is written and B is written. So A, my dear children, is the bigger cell and B is the smaller cell. So we first have to identify what is A and B. That is the easiest way to find the answers. Before reading the options, find it. So A is what? Since it is bigger cell, it is going to be vegetative cell. B is going to be what? Since it is smaller cell, it is going to be generative cell. Now read the option. A is the generative cell. Wrong, because A is what? Vegetative cell. A is the vegetative cell with abundant food reserve and B is the generative cell which forms the male gametes. A is the vegetative cell which has abundant food reserve, is it? Yes. What is the role of vegetative cell? To provide nourishment to the generative cell out there. So yes, they have dense cytoplasm, it has abundant food reserve just like that. And B is the generative cell which forms the male gametes. So I have got my answer, the option would be B. But still let me read the other option. A is generative cell, again wrong. A is vegetative cell which gives rise to male gametes. Vegetative cell does not give rise to male gametes, right? Gametes, G over here. Generative cell does that. So this is again going to be wrong. Your answer for this question is going to be B. Cool? Yes, ma'am. Okay. Now I have a homework question for all of you who are watching this video. Put down in the comment section that this is homework number 1 and this is the answer. Among the following statement related to pollens, choose the correct one. Statement 1. In 40% of angiosperms, pollen grains are shed at 3-cell stage. Statement 2. Intine is made up of cellulose and pectin and it is discontinuous with germ pores. Over here you have option. This came in KCET 2018. Tell me down the answers in the comment section below. You can just write A, B, C. First answer, first question, A, B, C, D, whatever is the option. Done? Cool? Let's go. So I hope until now it's super clear. Now we'll go ahead with what pollen as allergen and also a performance booster. So listen to me. Pollen grains are really tricky. They can cause allergies, they can cause asthma, they can cause bronchitis, they can cause respiratory issues as well. And the most common thing that causes all of this is Parthenium grass. Might have seen this. Okay, it's a wild thing that goes around. It's Parthenium, or you also call this as carrot grass. This is an invasive weed. We brought it from somewhere because of its beauty and now it's a weed. It does not allow other plants to grow. Okay, now it is highly nutrient rich. So we saw the bad part of pollen grains, it is allergen. The good part of pollen grains is that it is nutrient rich, because obviously they need to have nutrition to provide nutrition to the gametes out there, right? So it is highly nutritious, which is why it can be used at food supplements as well. And you know what? It is very popular in Western countries. The pollen products, that are tablets, such as syrups out there, they are very popular in Western countries. Ma'am, do we need to remember Western, Eastern and all? Yes. Yes, you need to. You can get question anywhere from your NCERT in KCET examination. So pollen products, my dear children, available as tablets and syrups, and they are mostly popular in what Western countries out there. Now some of the athletes and race horses, they also take this. Why? Because this is going to be a performance booster. It enhances the stamina. So it is allergen. Parthenium grass is one such example. It is the products of it, such as tablets and syrups, are very much famous in Western countries, and my dear children, it's a performance booster in the case of athletes and race horses. Now, pollen viability is one more topic. With that, we are going to end the male part of the plant. Pollen viability means what? For how much time will it remain viable, or will it be able to stay okay until it grows and germinates on the stigma? That is what you mean by pollen viability. So pollen must land on stigma before losing viability. Viability is what? The capacity to live, to grow and to germinate or to develop further for the process of what? Fertilization to happen. So viability definitely varies depending on the temperature as well as the humidity. There are few examples given in NCERT which is super important for you to remember. Remember, rice and wheat is something that we have as a staple food, right? So their viability, my dear children, is just 30 minutes. This is something very common to us, so we can remember. Rice and wheat, their pollen viability is just up to 30 minutes. So if within 30 minutes they are not landing on the stigma and things like that, they are going to lose their viability. That's all. So rice and wheat, just 30 minutes. But there are some things which have long-lived viability as well. For example, the plants belonging to Rosaceae, Leguminosae and Solanaceae. So Rosaceae as in rose, Leguminosae as in pea plant, and Solanaceae as in your chili and things like that, or your potato. Okay, so Rosaceae, Leguminosae, Solanaceae, remember their names. These say are basically going to be viable for what? Months. For months. Fine. Now the pollens can be stored also. Do you know that? Pollens can be stored, and this is known as cryopreservation. Whenever you are storing this in liquid nitrogen at minus 190 degrees Celsius, this is known as cryopreservation, and basically you can use this for crop breeding program. Later on, if I feel one plant is not there, it is extinct or it is going to be endangered, you can store them and take the pollen grains and fuse it with the female gamete out there and then make my things work. Okay, so please remember that also. So pollen viability is the ability to grow, germinate as well as do the process of fertilization. Some of them has less viability, 30 minutes only, for example rice and wheat. Some of them can be viable for months, Rosaceae, Leguminosae as well as Solanaceae. And pollens can be stored in minus 190 degrees Celsius, 196 degrees Celsius in liquid nitrogen. Cool? Yes. Fine. Now we go ahead with the next concept, that is what all about gynoecium. So the reason I am not stopping anywhere is that this is masterstroke, and masterstroke means we have to cover it fast. I do not want to waste any bit of your time. So if at all you want some time off, you can pause the video for some time and then listen to me, but I'll recommend you complete it altogether. But if you want to revise, pause and just go through revise. Do not make your notes right now. Okay? Fine. Let's go ahead with the gynoecium or the female reproductive organ. So androecium was the name of male reproductive organ. Gynoecium is the name of female reproductive organ. So over here, you also call gynoecium as pistil. Okay, you call this as pistil. You know, stamen was the other name for androecium. Here it is known as pistil, or you can also call this as carpel. Pistil or carpel. This is the name of gynoecium. And this gynoecium, my dear children, is made up of stigma, style and the ovary. Stigma, style and the ovary. And without wasting much amount of your time, I'll just draw a diagram and I'll show it to you how things work over here. So this is the stigma. Okay, this is going to be the style and this is going to be the ovary. Inside the ovary, you have the ovule. What is this? So first of all, this is the ovary and this is going to be the ovule. Well, this cavity that you see within the ovary, it is known as what? Locule. The ovarian cavity is known as locule. Cool? Now, ovule cannot just stand there like that, okay? Ovule is the place where the female gametes are going to be formed. So, obviously, we need to provide some nourishment. So, ovary decides to provide nourishment with the help of a structure which is known as placenta. This is going to be the placenta. And placenta is attached to the ovule with another structure which is known as funicle. So placenta is there attached to this ovule with the help of funicle, and the point of attachment which is there, it is known as hilum. It is going to be known as hilum. Okay, and this ovule, my dear children, is feeling very cold inside the ovary, which is why it needs two sweaters. So one sweater is there, this is known as inner integument, and one more sweater is there, you call this as outer integuments. What are this? So these are inner and outer integuments. And if at all you have seen, I have not closed this integuments. There is an open space right there. What do you call this open space? This open pore-like structure is known as micropyle. It is known as micropyle. And whenever micropyle is there, wherever micropyle is there, you call this as micropylar end. And opposite of micropylar end will be known as this end. This end is known as chalazal end. What do you call this? Chalazal end. So you have micropylar end. Opposite of wherever the micropyle is, it is known as chalazal end. And remember, always, everything important is going to happen in the micropylar region. Everything important is going to happen in the micropylar region. So over here, you have the ovule. I spoke about it. Ovule has integuments and everything out there. What is there inside the ovule? What is going to be there inside the ovule? So inside the ovule, my dear children, you would be having the cells of the nucellus. So nucellus is again a tissue just like your sporogenous tissue, no? So nucellus is also a tissue made up of multiple cells. And out of them, one cell is there. One cell, my dear children, is there. I'm making a throne over that particular cell. And this particular cell which is there is known as MMC. Ma'am, what? MMC? You told MMC is microspore mother cell. Here it is known as megaspore mother cell. You remember in sporogenous tissue, each and every cell was responsible for undergoing meiosis and form the microspores out there. But over here, it's not like that. Even though you have multiple cells in the nucellus, they are there for nourishment. Only one cell will be there, only one cell, which is known as megaspore mother cell, which is going to undergo division. That is one very important difference that you have. And also one more thing, ovule over here. The name of the ovule which is there, ovule is also known as Megasporangium. Megasporangium. So there also, what did you have? You had Microsporangium. Inside the Microsporangium, we had sporogenous tissue, right? Here inside the Megasporangium, you have what? Cells of the nucellus. And inside that, basically one cell will be there that is known as Megaspore mother cell. You relate and you learn, you will never forget. Okay, so ovule is also known as Megasporangium, and over here you have nucellus. One cell is known as Megaspore mother cell. Is it clear? Is it clear? So first of all, let's decode it. Ovary is there. The cavity within the ovary is known as locule. Over here you have placenta, which is going to provide nourishment to the ovule. It is attached with the help of funicle. The point of attachment is known as hilum. It has two integuments, inner and outer integument. At one area it is not covered. This is known as micropyle. Why is this area not covered? Because this is where the pollen tube is going to come and get inside the ovule. Because inside the ovule, what is there? Inside the ovule you have cells of the nucleus, and also you have megaspore mother cell, which is going to divide and form something that is known as megaspore, and later will form the female gametophyte. There we form the male gametophyte. It was known as pollen grains, right? Here we form the female gametophyte, which is known as embryo sac. So everything is going to happen up over here. Everything is going to happen up over here. Micropylar end, opposite of is known as chalazial end. Now one very important piece of information. It's not like every time the placenta and the funicle and everything is going to be here. It can be at this location also. Whenever it is ulta, you call this as anatropous ovule. Anatropous ovule. Whenever suppose this and this structure, the placenta and funicle is over here, it is known as anatropous ovule. And also understand, at this particular time the chalazial end remains here only. It's not like wherever placenta is there, only chalazial end will be there. No. Where will be the chalazial end? At the opposite side of the micropyle. Wherever the opposite side of micropyle is, there you will be having the chalazial end. Okay? Cool? Fine. So, stigma, style and ovary we studied. Ovarian cavity is known as locule, which in turn contains the ovule, which is known as megasporangium. Yes ma'am, we know about it. Now we studied about this also. Ovule, megasporangium arise from the placenta inside the ovary. Placenta is located inside the ovarian cavity, we know. Funicle is what? Stalk attaching ovule to the placenta. Hilum is the junction between funicle and the ovule. Integuments, one or two protective layers which are there. And micropyle is the small opening at the tip for the pollen entry. Chalazial is the opposite side of the micropyle. And nucleus is the mass of cell with reserved food material out there. Done. Now as I told you, sometimes over here you see the ovule is there, inner and outer integument is there. This is the micropyle, and here you have the funicle. This is the funicle. Here you will having the placenta as well. That is not being shown over here. So please understand, this is ulta structure and this is known as anatropous ovule. This is known as anatropous ovule. Done. Now my dear children, there are certain pictures given in NCERT which you ignore, but these should not be ignored. Okay, this is important for you guys. So, there are two conditions that we have. That is, we can have multiple carpels. That means multiple female reproductive part. And all these carpels having stigma, style and ovary. Basically, suppose their stigma and style are separate, but ovary is one. They all have one ovary. What do you call this condition as? This condition known as syncarpous condition. Whenever, suppose you have multiple carpels out there, okay, there are multiple carpels out there, but all the carpels ovary are fused together. This is known as syncarpous condition, and this is what you see in Papaver somniferum. You know, Papaver is a drug, Papaver is a plant that gives you the drug which is heroin as well as morphine. So if at all you see the structure, it's given in your NCERT. Here you have shown the stigma and the style. So stigma and style are over here, but they have a syncarpous ovary, as in all of them have a unified ovary. So this is the stigma as well as the style. Now if at all you see inside, there will be only one ovary. This condition is known as syncarpous condition, and this is what you see in Papaver. Now opposite of it can also happen. You can have multiple what do you call carpels out there, and none of them are fused. All of them have their own ovary out there. This is known as apocarpous condition. An apocarpous condition is seen in Michelia. You might have seen this particular flower. We call this champak in Hindi, chembagam in Malayalam. Okay, so this has a very nice smell. Okay, so this particular flower, if at all you open the petals and all, it looks like this. They have multiple carpels out there, and the picture given in your NCERT, it looks like this. So there are multiple carpels, my dear children, and also understand they will be having multiple ovaries as well. This condition is known as apocarpous condition. Okay, so Michelia which is there, it is apocarpous, and Papaver which is there, it is syncarpous in condition. Done? Yes. Okay. So you can remember Papaver and syncarpous. PS5 you might be playing PS, right? So you can remember like that. Papaver is syncarpous in condition, and Michelia, my dear children, is apocarpous. Ma. Okay, Ma and PS. Fine? Cool. Now, there is one more thing. Some plants will be there which has multiple ovules. Inside the ovary, there can be multiple ovules. Well, you know what? Well, do you know one very important thing? That is, suppose fertilization has taken place, male and the female gamete fused, right? So the ovary which is there, it is going to form the it forms the fruit. What happens to the ovule? Ovule will be forming the seed. So suppose you are seeing a fruit which has multiple seeds, just understand that multiple ovules are there. So papaya, watermelon are examples. This you can easily remember. Orchid is something for which you might have not seen the flower, but please remember, orchid which is there, it has multiple ovules. Orchid also is among papaya and watermelon, and they also have multiple ovules. On the other hand, if at all you talk about wheat, paddy and mango though you have seen, no, only one seed is there, right? So they just have a single ovule. Even wheat, even paddy, everything, my dear children, all of them has single ovule only. Okay, so we spoke about the structure of the gynoecium. We spoke about Megasporoangium as the ovule and what all thing is there inside the ovule. We spoke the condition where multiple ovules are there, that is this, where only one ovule is there, that is this. We spoke about the condition where multiple carpels are there and all of them are fused, which is known as skin carpus condition. We spoke about the condition where all the carpels are there but they are not fused. We call this as apocarpus condition. Now we are going to deep dive into what? The structure, the actual structure of ovule and what exactly is happening inside the ovule. It is known as Megasporangium, right? So obviously we need to go ahead with Megasporogenesis. We need to make that megaspore mother cell to basically divide and give us what? Megaspores. This megaspores which is there will give us the female gametophyte. There, Microsporogenesis, microspores and male gametophyte, right? Here also it is going to give us the female gametophyte. But things are going to be quite different. So let's see what is going to happen over here. So here, my dear children, megaspore mother cell undergoes division. Obviously, if it was diploid over here, it becomes haploid here in the first meiotic division. In the second meiotic division, again, you get 4 cells out there. So, ma'am, again, do they form megaspore tetrads? No, megaspore tetrad is not there. 4 megaspores are being formed. But you know what? One very sad thing happens. 3 of them are going to get degenerated. 3 of them get degenerated, only 1 cell is going to be there. And this 1 cell which is there, it is known as the functional megaspore. What do you call this? The functional megaspore. This is the only cell which will be further forming the female gametophyte. It further forms the female gametophyte, and female gametophyte, it is also known as the embryo sac. You also call this as what? The embryo sac. So female gametophyte, or you call this as embryo sac, and this is going to be what? The functional megaspore that is going to form this. Only 1 megaspore. Since only 1 megaspore is functioning over here and forming that, you call this development as monosporic development. What do you call this? Monosporic development. Only 1 spore develops. It's not like microsporogenesis, right? Here things are going to be quite different. So undergoes meiosis producing 4 megaspores. Only 1 functional megaspore will be there. 3 of them are going to degenerate. This is known as monosporic development that is going to happen to form the female gametophyte. So everything is happening inside the ovule. You need to remember that. Nuclear cells are there, 1 cell divided and form the megaspore, and this megaspore is now going to form the female gametophyte, which is also known as the embryo sac, and we are going to see about that, what exactly is going to happen. So see, this is that functional megaspore that you are having. It has its own nucleus, agreed? So what exactly happens is that this nucleus is going to divide thrice. How many times? Thrice. Okay, you call it 3 mitotic division. 3 mitotic division is going to happen up over here. So I'm writing it. Do not forget 3 mitotic division. Okay, so first mitosis happens, you get 2 nucleus. Remember, cell wall formation is not happening. The nucleus is the only thing that is dividing. You got 2 nucleus, then again this divided, you got 4 nucleus, then again these divided, you get what? 8 nucleus. How many nucleus? 8 nucleus you got. And then you know what? After this nuclear division, you call this division as free nuclear division. Free nuclear division. After this division, slowly cell wall comes around cell wall is going to come around these particular nucleus out there. and you will get certain cells which are going to arrange themselves to form what? The embryo sac. So for now it was the functional megaspore. It kept on dividing, and now when the cells and everything is arranged together, you will now call this as what? The functional embryo sac out there, the female gametophyte. So over here, let's see how is this happening. So 3 of the cells along with the nucleus go at the top and get accumulated over there. They are known as antipodal cells. Antipodal cells are found in the chalazal end. The chalazal end. 2 of the nucleus get surrounded by cell wall, and now they are called as synergid cell. One cell which is out there, which is the most important cell out there, this is the female gamete, or you also call this as the egg cell. This is the only egg cell that you have. So in the embryo sac, there is only one female gamete. Got it? Together, synergids and egg cell, they are best friends. You call them as egg apparatus. You call them as the egg apparatus. Okay. Now, this whole thing that you see, this whole thing that you are able to see, it is going to be a very big cell. And this very big cell will be having what? 2 nuclei out there, 2 nuclei. So this is known as the polar nuclei. 2 polar nuclei will be out there. My dear children, when we underwent meiosis, always remember all of them were haploid, right? So the nucleus, everything is haploid in nature. All of them are going to be having half the number of chromosomes in their nucleus. But now if at all you ask, 8 nucleus were there. Are there 8 cells over here? No. 3 cells, 3 cells, 3 plus 3, 6. This is one big cell, that is the 7th cell. 7th cell has 2 nuclei. So what do I call this condition? This condition is known as 8 nucleate, 7-celled condition. 8 nucleate, 7-celled condition, and all of them are haploid except this cell. What do you call this cell as? The cell is central cell. And why is it not haploid? Baba, it has 2 nuclei, 1 nuclei having haploid, another nuclei also having haploid. Haploid, it becomes what? Diploid in nature, 2n in nature, n plus n, 2n. So this is the only cell which is diploid in nature, and that is your central cell. I hope this is super clear to you. Is this clear? This is super important. Okay, and everything, egg apparatus is going to be there in the micropylar end. The pollen tube carrying the male gametes also come in the micropylar end only. Everything important, even this division which is happening, the Megasporogenesis which is happening, that also happens in the micropylar end. Done. I hope this is clear, right? Now I'll go further and explain you one more thing over here itself, and that is double fertilization. Since we anyway talking about all this thing, we can talk about double fertilization also. So my dear children, here it is free nuclear division, 3 mitotic division happens, later cell wall. So cell wall is not being formed while the nucleus is dividing, later on cell wall formation happens, and then you get something like this. There is egg apparatus, there is antipodal cells, there is central cell with 2 polar nuclei. This is the only diploid cell out there. Everything else is haploid in nature. Cool. Now my dear children, suppose the pollen tube came. Okay, suppose the pollen tube came. It comes like this. And the pollen tube, my dear children, will be having 2 gametes. We discussed it will be having 2 male gametes, right? So what exactly happens? You need to guide the pollen tube inside. Okay, and who does that? Synergid cells does that. Synergid cells has a very important thing over here. It's like finger-like projection. You call this as filiform apparatus. What do you call this? Filiform apparatus. And what do? They basically guide the pollen tube inside the the egg apparatus out there. So while this happens, you need to understand one very important thing. There are such thick friends, no? Synergids die for the egg cell out there. Basically, when the pollen tube is formed, it goes through one of the synergids, which is why it gets degenerated. It gets degenerated. Now you listen to me carefully. One male gamete out there, one male gamete with the nucleus out there, which is having n number of chromosome, here also n number of chromosome, they are going to fuse together. The male gamete and the female gamete, each of them having haploid number of chromosomes, are going to fuse together to give rise to zygote. This fusion is known as, and zygote is what? Diploid in nature. zygote is going to be diploid. Why? Because N and N, it becomes 2N in nature, right? And this fusion is known as syngamy. This is the first fertilization that we have. This is known as syngamy. Now, I told you one more male gamete is there. What happens to that? This male gamete will be coming with haploid nucleus and fusing with the two polar nuclei, or basically the central cell out there. It fuses with the central cell out there. The second male gamete, having haploid number of cell, fuses with the central cell out there, having diploid nature. Now N plus 2N, it will give rise to what? 3N. And what is this structure which is being formed? Here you called it a zygote. What do you call this as? You call this as primary endosperm cell. What do you call this as? You call this as primary endosperm cell, which is triploid in nature, not diploid, not haploid. It is triploid in nature. And what do you call this fusion as? You call this fusion as triple fusion. Why triple fusion? Because 3 nucleus are fusing. 2 polar nuclei and 1 nucleus of the male gamete. So which is why you call this as triple fusion. Now you told me fertilization is happening twice. Yes, dear children, which is why this is known as double fertilization. I hope you will remember this. So haploid, male gamete, female gamete forms zygote. It is known as, it is diploid in nature, and this process is known as syngamy. Over here, the male gamete as well as the central cell, which is diploid in nature, fuses together to give a triploid structure that is known as primary endosperm cell, and the nucleus will be known as primary endosperm nucleus. This primary endosperm cell, my dear children, later will be giving us a tissue that is known as endosperm tissue. What is this endosperm used for? Endosperm will be useful for nourishment of this zygote. That is why we need endosperm. Endosperm nourishes the zygote such that the zygote, which is single cell, divides and form an embryo and later form the whole plant out there. So endosperm is there for nourishment. And always remember, endosperm is something that is going to form first. It is something that is a primary endosperm cell, divides, forms an endosperm tissue first, and then only zygote will be basically forming the embryo later on. Okay, that's all. Fine. I hope this is super clear. So we'll come back to the embryo sac structure which we had. So nuclear division, functional megaspore nucleus divides, 2 nucleate stage, then 4 nucleate stage, then 8 nucleate stage. I told you 3 division happens. No immediate cell wall formation happens. This is known as free nuclear division. Cell formation, after 8 nucleate stage, cell walls form, organizing the embryo sac. Egg apparatus is there at the micropylar end, 1 egg cell and 2 synergids with filiform apparatus for pollen tube guidance, right? Central cell contains 2 polar nuclei, antipodal cells at chalazal end contains 3 cells, and mature embryo sac is 8 celled in nature, 8 nucleate in nature, but 7 celled in condition. This is the picture that you have in your NCERT. Got it? Yes. Cool. And with this we discussed it, so let's go ahead to the next thing, that is the types of pollination. So do we need to do this first, or let's go ahead directly with something else? So just give me a second, okay? Just give me a quick second. We will talk about this first, then we will come to that. Pollen-pistil interaction. Since anyway we are talking about fertilization, double fertilization, all of it, let's talk about this pollen-pistil interaction. So my dear children, I already told you that basically the pollen grains have to land on the stigma, and then it will be forming the pollen tube. But this is not an easy process. There are some chemical signals happening over here. It can be either inhibiting the growth of the pollen tube or can be promoting it. So there should be some chemical recognition happening, then only it will be going ahead. If at all the pollen tube, pollen grains are compatible, now it will form the pollen tubes. Okay, it will form the pollen tube. And always remember, pollen tube formation does not mean that fertilization will anyway occur. There are some plants where pollen tube formation happens, but it does not reach the ovule. So pollen tube formation does not guarantee any fertilization. You should need to know that as well. Okay. So after compatible pollination, the pollen grain germinates on the stigma. It produces a pollen tube through one of its germ pore. Yeah, it forms the germ pollen tube through its germ pore. Now, In plants where pollen is shed at two-celled stage, the generative cell divides during the pollen tube growth to form two male gametes. I told you 60% of angiosperms shed in two-cell stage. Generative cell and vegetative cell is there. This generative cell will now divide when the pollen tube formation is happening. That's when it will be dividing into the two male gametes out there. In plants with three-cell stage, the pollen already contains the male gametes, so they just flow down. Now, the pollen tube enters the ovule through the micropyle. It then enters one of the synergids, I told you, with the help of filiform apparatus, which guides the pollen tube entry into the ovule. So, filiform apparatus will be helping in guiding the entry towards the ovule out there. That's all. And one synergid will lose its life. Cool. Now, over here, I already spoke about triple fusion, but I can make you read about it. Sorry, triple fusion and syngamy. So, first fertilization happens over here. One male gamete fuses with the egg cell. This fusion is known as syngamy. It forms what? Diploid zygote. So, diploid, haploid, triploid, all of it is super important. Okay, it forms the diploid zygote out there. Now, triple fusion, the other male gamete fuses with the two nuclei in the central cell. This fusion of three nuclei, two nuclei, polar nuclei, and one nuclei of the male gamete is known as triple fusion. Okay, it is going to form what? A triploid primary endosperm nucleus, and the cell which is having this nucleus is known as primary endosperm cell. Fine? Fine. Now, double fertilization, because I told you, since both syngamy and triple fusion occur in the same embryo sac, it is known as double fertilization. It is a unique feature of flowering plants only. Now, what happens after fertilization? What is going to happen after fertilization? That is also super important. So, after fertilization, my dear children, the central cell after fertilization becomes what? Central cell after triple fusion becomes the primary endosperm cell, I told you. Yeah, PEC, primary endosperm cell. It develops into endosperm tissue. These cells are going to divide further and basically going to form what? The endosperm tissue out there. And always remember, endosperm forms first, because this endosperm is responsible for basically helping the zygote to develop into an embryo. Zygote is a single cell. Embryo is multicellular in nature. And who provides the nourishment? This particular endosperm will be providing the nourishment for that. Okay. And over here, I have a question. So, in apple, the chromosome number in the gametes is 17. What is the chromosome number in its primary endosperm nucleus? What is the chromosome number in the primary endosperm nucleus? So, number of chromosomes in the gametes, it's 17. Okay. Now, we are asking about the primary endosperm nucleus. Primary endosperm nucleus, we all know, it is triploid in nature. 3N in nature, right? 3N in nature, triploid in nature. So, if N in gametes, my dear children, we have haploid thing. Gametes will always contain haploid only. So, over here, if N is equals to 17, how did I get that? Because over here clearly it's mentioned chromosome number in the gametes is 17. In the gamete, obviously it is going to be haploid, the least amount. So, N I am assuming it is 17. So, what is going to be 3N? So, 3 into 17, if I do, you will get your answer. What is the answer? Your answer is going to be 51. Your answer is going to be 51. Got it? Now, over here, I have a question for you guys to solve. If a tetraploid plant contains 48 chromosomes in its nucellus is a normal cell out there. If a tetra means what? 4N. Tetraploid plant contains 48 chromosomes in its nucellus. Okay, in the cells of a nucellus, it is 48 chromosomes, and I am already saying this is condition where it is 4N. It is 4N in nature. Then the number of chromosome in the egg cell and in the synergid respectively will be what? In the egg cell and the synergid, what do you think the answer for this question will be? Any idea? Any idea? So listen to me carefully. I told you, if something is having 2N, it becomes half. It becomes N in nature. If it was 2N is 32, then half of it will be what? 16 in the case of gametes, right? So over here you are asked a similar question. They have just twisted things. They told there is a tetraploid plant which is basically having 48 number of chromosomes out there. So what is the number of chromosomes in the egg cell, that is in the gamete, and also in the synergids? Gamete, synergid, everything are haploid in nature generally, will be having half of it only. So whatever this number is given over here, just have the half of it. You do not need to do anything else. just have the half of it. If 4n was the normal thing, then gametes will be 2n in nature this time. It will be half of it. So 48's half is what? It is going to be 24. In this particular condition, your answer is going to be, you're asked about X cell and the synovids. Both of them are going to be 24, 24. I solved the question. It was a homework question, but I solved it because I thought it would have been a little bit tricky. But I will give you a question. The question is, okay, the question will be, okay, what is the number of chromosome in the primary endosperm nucleus? Here in this condition, what is the number of chromosome in the primary endosperm nucleus in this particular condition? Try it out and let me know. Works? Cool? Yes. Now we'll go all about the endosperm as well as the embryo. So I spoke about that endosperm development precedes and things like that. We need to know more about it, right? So what is the role of endosperm? To provide nutrition to the developing embryo. Process. So primary endosperm nucleus which was there, the nucleus which was there, it divides repeatedly. It keeps on dividing, just the nucleus. Okay, the nucleus keeps on dividing, and it keeps on going. Forms triploid endosperm tissue. When does it form triploid endosperm tissue? Listen to me carefully. So the nucleus has divided, right? Some nucleus will be covered by cell wall. If yes, you are making cells out there. These cells will come together to form what? Tissue. What is this tissue now? You call this as endosperm tissue, or basically you just call this as endosperm itself. That's all. Some nucleus will not be surrounded by cell wall, and you call this as the free nuclear endosperm. The free nuclear endosperm. So PEN undergoes nuclear division without cell wall formation, and that is known as free nuclear endosperm. Where do you see it? You see it in coconut water. The coconut water that you see is just the nuclei, is just the endosperm nuclei that is there. It is not covered by the cell wall. If at all you keep it for a long time, you might be seeing that all these nuclei are being covered by cell wall, and it forms the white kernel that we eat. So this white thing that we eat is nothing but the cellular form of endosperm. The nuclear ones were there, the water, slowly they form the cell wall around it, they become cellular, and that is what we eat. It is known as the cellular form of endosperm. Remember that. But anyway, we spoke about free nuclear endosperm and the cellular form of endosperm. What is going to happen with this endosperm later on? What is this fate of the endosperm? So my dear children, it can be completely consumed before seed maturation. It can be completely consumed before seed maturation. As in, what is seed? Ovule, which has undergone fertilization, will now be known as a seed. Ovule, which has undergone fertilization, is now known as seed, right? So, when the zygote transforms into an embryo, at this particular point of time, all the endosperm which is there is an option that the endosperm which is there, it is completely used up for the formation of the embryo. That is one condition. Okay, there is a condition where the endosperm is not completely used up, still some amount of endosperm can be left. And you have two different names for it. Different type of seeds which is going to be formed. You have two different type of seeds. One is known as non-albuminous seed. Non-albuminous seed. What do you mean by that? Non-albuminous seed means it has completely utilized the endosperm. The embryo has completely utilized the endosperm for itself. There is no endosperm left behind. What does the embryo do with the endosperm out there? Embryo will form its own reserve food material. This is very important. Embryo did not want endosperm. It wanted to have its own reserve food thing. So what is that thing? It is known as cotyledon. So you might have seen there is embryo sac right there. You might have seen there are some plants which have two cotyledons. For example, chickpeas. Chickpeas, my dear children, if at all you notice, you see something like this in the center, and then two cotyledons are there. You can break into two. Right? So what are these cotyledons? They are the stored form of food which the embryo is made with the help of endosperm. So now they do not have endosperm at all because all the endosperm is converted into what? Cotyledons. So always remember one very important thing. The non-albuminous seeds which are there are found in plants which are having two cotyledons. They will be converting into two cotyledons. You call this condition as dicot. Okay, you call this as they are dicot in nature. Example, you have chickpea. Whatever you can break into two, all of them come under this. All of them are going to come under this particular thing. Okay, so apart from pea, your groundnut which is there, your beans which is there, everything, they come under dicotyledon plants. Now, there is one more type of seed which you can see. One more type of seed is there where some amount of endosperm is still remaining. You call them as albuminous seed. Albuminous seed. So here, Baba, endosperm is still remaining. Little bit endosperm is utilized also. So here, instead of having two cotyledons, they will be having just one cotyledon. They are known as monocots. Why they just have one? Because they have little amount of endosperm also persisting. Remember, they just have one cotyledon, so they cannot be broken into two. For example, if at all you talk about this thing, that is wheat, rice, barley, coconut, think of them, my dear children, they are monocots. Now, the next slide is all about it. That is the embryo. So before I go into that, so this is what we were discussing right now. So over here, my dear children, when I say that the ovule is transformed and a lot of things are happening over here in this particular condition, you need to know that ovule is transformed into what? A seed. And seed, my dear children, will be having seed coat. It will be having seed coat around it. How is the seed coat formed? Remember, ovule had integuments. These will now turn into the seed coat. These are now known as testa and tegmen. Testa and tegmen are basically the seed coats. Apart from this, what does this seed have? This seed can have either one cotyledon, the embryo, right? The seed is having embryo. So the embryo can have either one cotyledon or it can have two cotyledon. If it has one cotyledon, it will also have some amount of endosperm. If it has two cotyledon, there is no endosperm that is going to be seen in this particular seed. I hope you got it. Now, what happens is that sometimes within the ovule, some amount of nucellus might still be remaining. Nucellus was there. Usually it is being utilized for nourishment and things like that. Sometimes some amount of nucellus might still be remaining within this particular ovule. And over here you have the embryo being formed, right? So this nucellus which is still persistent and which is there for nourishment of the embryo, this persistent nucellus is known as perisperm. What do you call this? Perisperm. And perisperm is found in B square. B square means beetroot and, beet and black pepper. It is what? Beet and black pepper. In these two things, you are going to find what? Persistent perisperm. This is found inside the ovule. You have the nucellus which is still there out there. Now, one very important thing that is there, people get confused between pericarp and perisperm. Pericarp is something that is totally different. Pericarp is basically the wall of a fruit. So this is an ovule. Ovule is basically nothing but a seed. Seed has seed coat. It is inside what? Ovary. Ovary is now what? Ovary is now a fruit. And fruit will also have its wall. It is known as pericarp. The wall of the fruit is known as pericarp. Perisperm is different. Perisperm, endosperm, both of them for nourishment. You remember like that. Pericarp is the walls of the ovary or the fruit walls out there. Always remember that. And they are multiple nature. If at all you talk about that, suppose I make a mango right over here. Okay, so this pericarp of the mango will have three things. One is the epicarp, that is the outermost portion. Okay, then we eat the flesh of the plant, right? This is known as mesocarp. What do you call? This is the mesocarp. And then inside that you have something like this. It is known as the endocarp. It will be known as the endocarp. Inside the endocarp, Baba, you will be having the seed with the seed coat. Inside the seed, what do you have? You have the embryo. The embryo which can give rise to a whole new plant, which is why we say seed is super important. Okay, got it? So this forms the different part of pericarp. Epicarp, or you also call this as exocarp. You also call this as exocarp. Okay, then you have mesocarp, then you have endocarp. Inside that you have seed and the seed coats out there. Done? Yes. Now over here, when seeds mature, its water content is reduced and seeds become relatively dry. Okay, when the seeds mature, their water content is going to be reduced very much and they are going to be dry. Almost 10 to 15% moisture by mass happens. And the general metabolic activity of the embryo also slows down. When the seed is going to be mature, the general activity of the embryo is also going to, metabolic activity is also going to slow down. Done? Done. Now, I was talking about embryo and I said there are two types of embryo, right? Monocot and dicot embryo. And now we will be talking about them. So, the embryo when it forms, it has a normal stages, or it has a few stages which is common for all. The zygote will be there, a single celled thing. The single celled zygote is going to divide into two cell thing. Again, it will form something else which is known as globular embryo. Then it will form a heart-shaped embryo. Then it will form a mature embryo. Okay, mature embryo. So basically, zygote. Then you have proembryo. Then globular embryo, heart-shaped embryo and mature embryo. Ma'am, is this important? Yes. The flow of this is super important for you guys to remember. So, proembryo, globular embryo, heart-shaped embryo, mature embryo. So you can remember like this: zygote pro, globe heart pro. Okay, zygote pro, globe heart pro. So you can remember like that if at all you want. Otherwise, you just remember it naturally. Zygote, pro embryo, globular, then heart-shaped, and then mature form of embryo which is known as grow. Okay, done? Yes. So the zygote is dividing, and as I said, it forms two types of embryo. One type of embryo is known as dicot embryo. Everyone who is attempting PU2 board examination, you should know the diagram of both the digote as well as the monocot embryo. So dicot embryo goes like this. Embryo will be having an axis. Basically, this is known as embryonal axis. Embryonal axis basically means the things that is going to actually form the root and the shoot. Okay. So this lower part that you see, you call this as hypocotyl region. This upper part that you have, you call this as epicotyl region. Okay. Here at the tip, you have the radicle. Radicle is something that is going to give rise to root later on. And radicle is covered by something that is known as root cap, because you know it goes into the soil. So it needs a cap, it is known as root cap. Here you have the plumule. Plumule is the future shoot out there. It gives rise to the stem, the leaf and everything. Now for formation of the plumule ahead and the radicle ahead, you need some storage units. You need some reserve food. And what is that reserve food? It is known as cotyledon. So cotyledon starts from here. You have one cotyledon right up there, and there is one more cotyledon out there in the case of dicots. So dicots will be having two cotyledons. Okay, and just be here in the picture they have not mentioned, but it will be look like in this. It is known as the plumule. The lower region is known as radicle and root cap. This is hypocotyl, and this part just above is known as epicotyl. That is the only thing that you need to remember in the case of dicot embryo. Dicot embryo is super easy to remember. Now, if at all you think that this picture diagram is difficult, no, Baba, it is not. So I made it like that, but it is not difficult. So you make a smaller one over here, and you make like this. And you make like this. That's all. Okay, and then you mention they are the two cotyledons. This is the epicotyl. At the tip you have plumule. Here you have hypocotyl. Here you have radicle, and then the root cap. Done. This is what dicot embryo. Here the endosperm is completely utilized to form the cotyledons out there. Now what? You have the monocot embryo. Here things are going to be different. In the case of monocot embryo, again you have an embryonal axis. Embryonal axis is common to all. So embryonal axis will definitely be there, but you do not need to give space over here. Why? Because they just have one cotyledon. And this one cotyledon has a name also. This cotyledon is known as scutellum. It is known as scutellum. Here you have the epicotyl region where basically you have the plumule. Here you will be having the radicle, and radicle is covered by what? Root cap. Ma'am, you said endosperm is also there? Yes. Little amount of endosperm will also be persisting, so I make it like this. One cotyledon, little amount of endosperm. Ma'am, is this the same? Apart from this, there is no other difference? There is difference. So listen to me. Covering the plumule, you have certain things which is known as coleoptile. What is it? Coleoptile. And covering the radicle, okay, here you will be having something that is known as coleorhiza. It is known as coleorhiza. Okay, P over here for plumule, R over here for radicle, you can remember like that. So coleorhiza and coleoptile is also there covering each of them. It is a part of monocot embryo. So monocot embryo has just one cotyledon and rest endosperm will be there. Since endosperm is persisting in this type of embryo, you call the seed, basically the seed that contains this embryo, as albuminous seed. Cool. Wheat, maize, barley, castor, coconut, all of them. Now, in your NCERT, they have given you a picture like this, which is difficult for you to remember. But you got the concept right over here. Now we can make this picture easily. So if at all I see, I have to make a shark's mouth. So let's make a shark's mouth. Done. Now shark's hand is there, one hand. And basically we need to make shark's tail. Shark's tail have been made. Then we come back, here is another fin. Here is another fin. Got it? Now, make a structure like this, a V like this, and color it up. Again make a V like this and color it up. Cool? Join this and let it go inside this and let it come back and let it go to one of the tail and then let it come back and join back over here. Fine. Now listen to me. This is going to be your coleoptile. This is going to be your plumule. This is going to be your cotyledon, that is cutellum. Endosperm is not mentioned over here, so you do not have to do it. This is, where is it? This particular structure that you see is your radicle. Covering the radicle, you have one thing that is known as the root cap. Now you make one more triangle like this, and then you have formed what? The coleorhiza as well. And that's how you did it. Now, one cotyledon is there, another cotyledon also tries to form, but it is not able to completely form itself. It is known as epiblast, and that is being mentioned in your NCERT. It is what? The epiblast. It is a rudimentary cotyledon that tries to form itself. Done. This is all. This is the monocot embryo which you need to know. Cool. Now, going ahead, there is a question. In the given diagram, identify the parts labeled as A, B, C and D. Now this particular picture is also given in NCERT and you have to find what is A, what is B, what is C and what is D. Remember, over here, we all got an understanding that A and B looks like what? Coleoptile and coleorhiza. And this side will be what? Scutellum. Whatever is left behind is going to be what? The endosperm. So we have to see if at all of this are matching or not. So A, if you see, A is going to be coleoptile. Is it coleoptile? Yes. Let's go with D then. D is what? Coleorhiza? Yes, it is. In any other option, do you see coleoptile and coleorhiza, or do you see radicle as D? No, right? You do not see radicle root cap as D. It's pericarp, pericarp, pericarp. So obviously pericarp is what? I already told you, it's the wall of the ovary. So obviously that cannot be the answer. So I have A option with A as coleoptile and D as coleorhiza. B is cutellum, and over here they have mentioned C as pericarp. Can C be the pericarp? Pericarp is the wall of the ovary. Can C be the pericarp? But this is what? The seed. Seed can have thick walls. In the case of the seed can be covered by a very big ovary having thicker walls, or it can be dry also. It can be just dry without any flesh out there. In that particular case, the outer layer will be what? The layer of the ovary only, and this layer can be known as pericarp also. It can be known as pericarp. So not always that it will be fleshy. For example, in groundnut oil, do you think it's fleshy? No. The peel is, it does not, there is no fleshy mesocarp out there, right? There is no fleshy mesocarp out there. In that case, the C will be pericarp. Got it? So your answer for this question, if at all you are able to see this and the D option, you have halfway done. Not have to worry about anything. Cool? This is a question that came in your KCET examination. Fine. So we discussed about the seed. Now there are some advantages of seed that we need to discuss. That is, first advantage is independence from water. Pollination and fertilization do not rely on any form of water. Okay, that is one very important thing. Seed can germinate by itself if at all there is little water itself. Dispersal and colonization. Seeds help plants to spread to new habitats out there. Agreed. Food reserve nourishes the seedling until they start photosynthesis. Yes, because they have food reserve in the form of cotyledons or in the form of endosperm. Now, protection, hard seed coat is there to cover them. Genetic variation, sexual reproduction, which is giving you the seed, obviously it leads to variation. Now, there are two types of very important seeds that you need to remember. Okay, So seeds also lose their viability. Some seeds are there which do not lose their viability for years, and there are two examples given in the NCERT. One is Lupinus articus. It didn't lose the viability for 10,000 years. That's crazy, right? To 10,000 years, Lupinus articus, which is found in the Arctic tundra, it did not lose its viability. Then you have Phoenix dactylifera, which you also call as date palm. Date palm. It didn't lose its viability for 2,000 years. And it was found during the Dead Sea excavation in King Herod's time. In King Herod's time, during the Dead Sea excavation, it was found that Phoenix dactylifera's 2,000-year-old seed was still viable. So 2,000 and 10,000. Lupinus articus is the one which is having more, it is 10,000, and the date palm is the one which is having less, that is 2,000. Phoenix dactylifera is the name for that. Remember their names, the names are super important. Cool. Now we have one more thing, and that is the true fruit and false fruit, and also apomixis and polyembryony. What do you mean by true fruit and false fruit, and what do you mean by apomixis and polyembryony? Listen to me carefully. Sometimes the ovary, ovary is the one that forms the fruit, right? But sometimes it's not just the ovary, it can be something else also. So ovary lands on the thalamus. So this thalamus can also take part in the formation of fruit. Or the pedicel which is there, that also can take part in the formation of fruit. These type of fruits which are not just formed of the ovary, but also the other parts of the plant, flowering plant, that is thalamus or pedicel, these fruits are known as what? False fruits. False fruits. For example, apple, strawberry and cashew. Apple is made up of the thalamus also. So in apple, if at all you see this particular thing is given in your NCERT, the picture, the seed is there. The seed will be covered by the fruit. The fruit should be ovary. So yes, ovary forms the fruit, definitely. This part of this is ovary itself. But the most of the part is basically thalamus. So thalamus is also contributing for the formation of fruit out there. Okay, how do I understand that the ovary is there? Because they have clearly mentioned mesocarp, endocarp and everything. So until this particular portion that you have, the hard portion that you have, it is the ovary that have taken part. But thalamus also takes part in this. So please remember, why do you call this as false fruit? Because over here, in the case of apple, the thalamus take part. Similarly, strawberry and cashew are also made up of other parts of the flower. Cashew is made up of the pedicel. Pedicel means the stalk-like structure which is there. Fine, got it. Now you have a term that is known as parthenocarpic fruit. What do you mean by parthenocarpic fruit? Suppose, my dear children, you have ovary and you have ovule. So there is no fertilization that is happening up over here. If there is no fertilization, do you think seed will be formed? No, Baba. No seed is going to be formed. And if there is no fertilization, do you think fruit is going to be formed? It should not be formed, but here it is forming because some hormones are active, which is why the ovary is transforming into a fruit. But there is no seed in this fruit. There is no seed in this fruit. Remember, without fertilization, whenever formation of fruit happens, this process is known as parthenocarpy. And over here, you will not find seeds because fertilization has anyway not taken place. Why is this happening? Because some hormonal imbalance is taking up there. Okay, so parthenocarpic fruits developed from without fertilization, example banana, can be induced using growth hormones, and they are seedless fruits. They are what? Seedless in nature. You cannot have any seed out there. Got it? Yes. Done. Fine. Example is banana, and you can induce it also. You can induce it in natural environment. So for example, tomorrow I want some seedless variety of fruit, I can do that. Cool. Yes. Fine. Now we have apomixis and polyembryony. What do you mean by apomixis and what do you mean by polyembryony? Listen to me carefully. So, until now we studied that basically embryo sac is there, egg and the male gamete fuses to form the zygote. Suppose the egg cell which is there, it is already diploid in nature. It's not haploid, it is diploid in nature. Do you need any other male gamete? No. This egg cell can itself form the zygote, can itself form the embryo as well. This process or formation of zygote without fertilization is known as apomixis. This process of formation of zygote, or basically the seed, okay, when you are forming the seed, this whole thing is now going to be seed. Seed without fertilization is known as apomixis, and this is very commonly seen in the case of Asteraceae and grasses. Ma'am, do we need to remember it? Definitely. Asteraceae and grasses, my dear children, they show apomixis, and remember, they mimic sexual reproduction but is actually a form of asexual. They just like sexual reproduction. Why? Because a zygote is being formed and things like that. But do you think they are sexually reproducing? No, Baba. It's asexual. Why? Because only one parent is being involved, and because of that, you are going to get clones over here. You are basically going to get clones over here. Just how the female parent was there, similarly all the baby plants will also be like that. So it can be happening because of the diploid egg cell. Some problem was there, the egg was already diploid. So this diploid egg cell forms without reduction division, develops into embryo without fertilization also. Apart from that, there is also one more way. That is, suppose in the ovule, there is some amount of nucellus still remaining. You remember perisperm that is what there for nourishment? That is not this. Basically, some cells of the nucellus, they get inside the embryo sac. They get inside the embryo sac and form embryo right there. These cells are already diploid in nature. These cells are already diploid in nature. Now, when they get inside the embryo sac, these diploid cell can give rise to zygote. And not just zygote, they further form embryos. And they do not form just one embryo, they can form multiple embryos. And this is known as polyembryony. Polyembryony. Remember, apomixis can be happening because of this or this. If it is happening like this, basically you find multiple embryos being formed, which is why you also call this as polyembryony. Okay, polyembryony is usually seen in citrus fruits such as oranges, mango and things. This is also important. Citrus plants which is there, oranges, mango, you will be finding this. Apart from that, apomixis commonly is found in grasses and Asteraceae. Okay, parthenocarpy is formation of fruit without fertilization, apomixis formation of seed without fertilization. This is asexual reproduction, you are getting clones. And how can it be useful? It can be useful for the farmers who want all the features that is there in the plant to be exactly like that. Because farmers, my dear children, they have to pay money for producing hybrid seeds. Then seeds, when they grow, basically this plant can have some genetic variation out there. It will not be passing the same traits every single time in sexual reproduction. But if at all you want the plant to be exactly the same every single time, you can just make use of the apomictic seeds. These seeds will be having the exact feature of the parent plant, which is why this can be used. So importance, hybrid loses traits if grown from natural seeds. Apomixis prevents segregation, allowing farmers to reuse these seeds every single year. Okay, reuse this particular seeds every single year out there. That is, it is cost effective for them. It is going to be cost effective for them. So this is apomixis and polyembryony as well. Then polyembryony example is citrus fruits, for example orange, mango, things like that. Apomixis in general, the example will be Asteraceae as well as grasses. Now, we have officially completed the chapter, but we need to cover some very important thing that we have missed in between, and that is the types of pollination, as well as a few things, artificial hybridization, emasculation and all of that. So let's study about that also. So there are three types of pollination. I'll explain it to you. I do not need all of this thing. I'll explain it to you in an easy way. So there are three types of pollination. One is autogamy, and one is gytonogamy, and one is going to be xenogamy. So listen to me. These two are cross-pollination, and this is going to be self-pollination. What do you mean by that? So suppose you have a plant, you have a flower, and the flower has both the male as well as the female part. From here to here, the pollen grains can go within the same flower itself. This is known as autogamy, or you also call this as self-pollination. Now suppose over here you have two flowers on the same plant. Okay, from here to here pollination is happening between two different flowers but of the same plant. This is cross-pollination. This is considered to be cross-pollination. But do you think genetically you will get something different out there? No, because the flowers are from the same plant. So genetically it is like autogamy. Gynogamy, genetically if at all you're talking, it's like autogamy, but it is actually cross-pollination. It is not a true form of cross-pollination, because true form of cross-pollination means here you have two plants, and from here to here, if the pollen grains are landing between two different plants, it is happening. It is true cross-pollination, and that happens in the case of xenogamy. So here you get genetically different plants, here you not get genetically different plants. This is cross-pollination but not true, and this is self-pollination where autogamy is happening. For autogamy to happen, you need to make sure that the pollen and it should be stamen as well as the pistil should be there. That means the flower should be what? Bisexual in nature. Bisexual in nature. There should be synchrony between the pollen release and the stigma receptivity. The pollen should be released at the correct time. The stigma should also be receptive to receive it. Okay, pollen release and stigma receptivity should also be there, and also their position and everything should be very much similar. Then only autogamy can happen. Now, my dear children, listen to me carefully. Under autogamy, you have been taught about two types of flowers. One flower is where in the root of the rhizoid region you have closed flowers, where you have the female and the male parts. Then at the top region you will be having some open flowers like this. These type of plants which have closed as well as open flowers, they have separate names for this. The open flowers, the closed flowers which are there are known as clistogamous flowers. Clistogamous flowers. They are always closed. The open one which are there, they are known as chasmogamous flowers. In chasmogamous flowers, since they are open, and since they have both male and female part also, if at all they can show autogamy, they can show geitonogamy, they can show xenogamy also. They can show all type of pollination. But what about clistogamous flowers which are closed and which have male and female part? The closed ones will always show autogamy only. Only autogamy, nothing else. Okay, only autogamy. And over here, remember, there will be definitely seed forming out there. Seed setting is assured. You do not need to be worried about anything. Do you need any pollinators? No pollinators are required. Seed setting is assured, and you will find the same type of plants being formed out there. Okay, that is a good thing about clistogamous flowers. Remember, they are usually found in the roots or just above the soil. And the example for this, you have three examples: Commelina, oxalis, and viola. Cow. But cow ends with V over here. Okay, Commelina, oxalis, and viola, they have these type of flowers. Got it? So let's see that. So first is autogamous self-pollination, stigma. Pollen from the anther, stigma of the same flower, needs synchrony in pollen release and stigma receptivity. Types of flowers: viola, oxalis, commelina. They have chasmogamous and clistogamous flowers. They are open flowers, chasmogamous, clisto, C, L for closed. You can remember like that. Okay, and here a short seed set is there. And advantage is that you do not need any pollinators out there. Disadvantage is that there is no genetic diversity. This is the closed flowers that you can usually see. And as I said, it is mostly found towards the root region. They are always closed. Okay. Now, yes. Now, geitonogamy and xenogamy, we discussed about that. Pollen to stigma of another flower on the same plant. Functionally cross-pollination, but genetically light autogamy. Xenogamy is like true cross-pollination, brings genetic diversity. Now, agents of pollination. You have multiple types of pollination. One is wind pollination, one is water pollination, and one is your insect pollination. Wind pollination examples are simple. One is your corn cob and one is your grass. You need to remember it. Corn cob, that is this, and also grass. There are certain features given for wind pollination. That is, pollen grain should be light and non-sticky for easy wind transport. If at all they are not light, they will not be transported easily. So they have to be light and they should be non-sticky. If they go and stick somewhere, it will be a big problem. So these features can be asked for any PU to champion out there. So stamen which is there, okay, the stamen, the male part which is there, it should be well exposed. Why? Because wind should be able to carry the pollen grains then only. Okay, so it should be well exposed. What about the stigma? Stigma should be large and feathery. Why? To trap the pollen grains which are traveling through the wind out there. What about the flowers? The flowers can be many. Flowers can be many, but always remember they just have a single ovule. Why? Because pollination may happen, may not happen. They do not have to waste their energy in the formation of ovule. So flowers are many, and whatever flowers are there, they will have only one ovule per ovary. Remember this, one ovule per ovary. This is a feature of wind pollinated plants. And examples are corn cob and basically grasses. Corn cob and grasses. This is super important. In the corn cob, the thread-like thing that you see is nothing but the stigma and the style. They are there for trapping, what do you call, the pollen grains out there. Okay, got it? The tassels are nothing but the stigma and the style. This is wind pollination. Please do not forget it. Now I am increasing the speed because the time is almost up, right? Now water pollination is seen, okay, so it is rare in flowering plants. It is rare in flowering plants. Only 30 genera, mostly monocots. Remember this. This is also a very important question. This is very rare in flowering plants, and it happens in only 30 genus, and my dear children, mostly they are what? They are monocots. They are not dicots, mostly monocots. 30 genera, mostly monocots. KCET question. Okay, common in algae, bryophytes and pteridophytes, the lower plant groups. Algae, bryophytes and pteridophytes, they have it in common. In the case of flowering plants, that is angiosperms, it's not common, only 30 genera and mostly monocots. Now example, Vallisneria, Hydrilla and Zostera. Vallisneria, Hydrilla, and also you have Zostera. In that, Vallisneria and Zostera are something that you need to know even more better. So Vallisneria is something where the female flowers, my dear children, they will be floating. Okay, they are going to be floating. And the male pollen grains are being released like this. They float and they come over here onto the stigma. That is how things goes in Vallisneria. It's surface level pollination that is going to happen. In the case of Zostera, my dear children, or you also call this as seagrass, over here pollination is going to happen in the submerged condition. The pollen grains and everything are going to happen inside the water itself. They're going to come and fuse together. So for that, the pollen grains have to be ribbon in shape. They are ribboning in shape, unlike the normal shape that they have. So Vallisneria and seagrasses, you also call the seagrass as Zostera. Please do not forget it. Vallisneria, they show the surface level pollination in the water. Female flowers float and the male flowers or pollen grains drift towards them. Whereas in the case of seagrasses, Zostera, pollination occurs underwater in the submerged condition. Pollen is long and ribbon-like. Remember, the pollen grains which are there in the water, they will be covered by the mucilaginous covering. And one very important question. Water lily, water lotus. Water lily and water lotus, do they show water pollination? No, Baba. They do not show water pollination. No chance. They are going to show what? They are going to show insect pollination. So that is most deceiving. Water lily and water lotus, they do not show water pollination. They show insect pollination out there, or wind, but not water pollination. Cool? Works? Vallisneria, will you forget? And Zostera. Zostera is also known as seagrass. Okay? Done. Now you have insect pollination, animal or insect pollination. So animals can be bees, butterflies, flies, wasp, ants, moth, birds, bats, primates such as lemur. Lemur is also a pollinator, remember. And rodents, reptiles such as geckos, lizards, they are all, my dear children, pollinators. So flower adaptations which are there, they are large, they are going to be colorful in nature so that the animals or basically the insects can be easily spotting them up. Insects cannot see colors, but yes, they can see certain things out there. Okay, which is why they need to be colorful and fragrant and very large. Small flowers are there clustered in inflorescence for visibility. Small flowers will be there, but they are clustered together to form inflorescence just for be visible. Fly and beetle pollinated flowers, they will be having a very bad odor such that it can attract the flies out there and the beetle out there. Now the pollen grains over here are going to be sticky. Why? Because these pollen grains have to stick onto the surface of the insect. Now when the insect goes around, the pollen grains will also be moved around with the help of that. So these are the features of what? Insect pollinated flowers out there. Now one very important thing, insect pollinated flowers will also give them some rewards. The reward can be nectar. Okay, the reward can be pollen grain itself. Some pollen grains will be eaten up, some pollen grains will be transported. And the reward can be shelter. 3 rewards can be there. And the most common reward which is there is providing safe places for egg laying, as I said, shelter. And this is found in Amorphophallus. This is found in Amorphophallus. Over here, if at all you see, this very big flower that you see is of Amorphophallus. It's like 2 human beings are there, it's more bigger than that also. Okay, here you find safe space for egg laying as well. Apart from that, in your NCERT, one more example is given, and that is of yucca and the moth. So yucca is this plant which is there, and moth is white colored moth. Here also, yucca basically provides safe space for egg laying, whereas the moth will be helping in the process of pollination. Got it? So there's an obligate relationship. Both of them, they stay together only, they survive together only, yucca and the moth. And this is Amorphophallus. I hope you got it. Okay, so yes, now what do you have? Just a second. Outbreeding and emasculation. This is going to be the last topic out there, and with this we will finish. So before I go ahead with outbreeding devices, I want to come here talk about artificial hybridization first. Artificial hybridization is a process where basically you want to produce your type of plants. Okay, your desired type of plants. What do? Suppose I have a plant which has all the good features out there, and I have another plant which also have some good features. I take the pollen grains from those plants and put it on the stigma of this flower, species specific only, and then I do the process of fertilization. Then I give all the atmosphere for the process of fertilization and everything to happen smoothly. This is known as artificial hybridization, or it is also known as controlled pollination, because it's not happening naturally. The pollination, at least, it's not happening naturally. You are doing it. You are helping it do it. Okay. So over here, suppose I have a flower which has male as well as the female part. Now male part will be releasing pollen grains which can land on the stigma, and that is a risk for us. Why? Because we do not want that to happen. We want to put pollen grains from some other plant. So what do? We do the first step, that is known as emasculation. We remove the male parts from here. If at all it's a bisexual flower, if it has male and the female part, you remove the male part from this. This process is known as emasculation. Okay, emasculation will be done. After that, what do? You basically cover this particular plant. Why? Because you want to make sure that the stigma is receptive. You make sure you wait for some time, the stigma is receptive. Later on, you take the pollen grains from the plant you want, and then you open this bag and you put it on the stigma, and then you rebag it. Bagging is done, putting of pollen grain is done, and then you rebag it. After rebagging it, you tag it that this is what I have done, this from this pollen grains from this plant and this from this plant, and I tag it and I keep it out there. Okay, and I wait for the process of fertilization to happen. All of it, am I not doing it? Yes. The plant obviously will undergo fertilization and everything, but rest everything is being done by us, which is why it is known as artificial hybridization. Remember, a question came in previous year PUC board, and that is controlled pollination. If there is basically crop improvement program where the farmers want to produce hybrid type of plants, which is the process that you are going to use. It is basically artificial hybridization which is going to be used up there. So you have to explain like that. You got it? It's very, very important. So emasculation, bagging, rebagging and tagging will also be there. Rebagging and tagging. Okay. Now if the flower is unisexual, you do not need to do emasculation. If the flower just has a female part, why do you need to cut the male part down? You do not need to do that. Okay. So that is artificial hybridization. Now with the last topic of today, and that is outbreeding devices. What do you mean by that? So generally we do not want the plants to do self-pollination, because the same features is going to get transferred. And self-breeding causes what? Self-pollination causes inbreeding depression. That means the plants have their toxic additives again and again, and it could lead to depression in plants. They might be not really good in surviving better in the normal environment out there, and a lot of things. Which is why plants have developed their own ways to avoid self-pollination, and that is known as outbreeding devices. Okay. One such is time difference. Basically, pollen release happens, but the stigma is not receptive. So even the pollen grains is land, the stigma is not receptive to take that up. That is one thing. Time difference. Second thing is spatial separation. Spatial separation means, suppose the female one is over here, the male is down over here. Do you think the pollen grains can go up there? Very difficult. So spatial separation is the second thing that you have. Third thing is self-incompatibility. This is a genetic mechanism where basically suppose the pollen grains land on the stigma, still there won't be some proper chemical communication happening up there, which is why pollen tube growth is inhibited. Pollen tube growth is inhibited. This is completely a genetic mechanism that is happening. Genetic mechanism preventing self-pollen from fertilizing the ovule is known as self-incompatibility. So time difference, okay, then spatial separation, then self-incompatibility, and now is formation of unisexual flowers. What do you mean by that? Formation of unisexual flowers means, suppose a plant just has the female flowers, and the plant does just have the male flowers. So obviously female flowers cannot go and fertilize up there, right? You need a male flower. So cross pollination is definitely going to happen. If something like this is going to happen, male and female flowers are going to be on two separate plants, obviously cross pollination will be happening, genetic variations will be there up there, proper sexual reproduction happening up there, right? Yes, this is one thing. Okay, unisexual plant only. This condition is known as dioecious nature. This is dioecious plants. What do you call these plants as? Dioecious plants. Where do you see it? You see it in papaya. Okay, in your NCERT, this example is given, you have to remember it. In papaya, there is plant which is having just the female flowers, and plant will be just having the male flowers. So both of them obviously have to undergo proper true cross fertilization or cross pollination or xenogamy to undergo the whole process. You get it? This is dioecious plants, and example is papaya. Now you have monoecious plants. Monoecious plants means what, my dear children? Monoecious example is castor and maize. Castor and maize, CM, Chief Minister. Okay, so over here in castor and maize, what do you find is, you have the female flower, you have the male flower. Both the sexes are on the same plant, but the flowers are different. So this is also unisexual flowers. This is known as monoecious condition. So here what can you prevent? Here you can only prevent one thing, and that is autogamy. Autogamy, because self-pollination can be prevented, but gynoecamy will still happen. Gynoecamy can still happen. So monoecious plants, my dear children, such as castor and maize. Castor and maize, they basically would not be helping much with outbreeding, because anyway gynoecamy can happen, but at least autogamy can be prevented. Now a question comes that monoecious plants, what all do they undergo? Autogamy is definitely not there. Do they undergo gynoecamy? Yes. Do they undergo xenogamy? Yes. From here the pollen grains can land on some other plant also, that is xenogamy. So xenogamy and gynoecamy is possible. Autogamy is not possible in the case of monoecious plants. Works. Yes. Castor and maize is the example for that. Cool? Dioecious, it's papaya. I showed you the picture as well. So this is the female flowers, and that is going to be the male flowers. Okay? Yes, and this is castor, my dear children, where the flowers are, as I said, male and female flowers are there on the same plant itself. There is no two separate plants. Got it? Yes. So I think with that, we have ended up everything which is there, and yes, and I hope that this particular video was super helpful for you. If it was, definitely let me know, and if at all you have any doubts, definitely let me know about that. And also please do the homework and come and be there for the live session which is going to happen on Saturday, okay, at 6 p.m. Yes, see you really soon. Lots of love from Jasmine ma'am. Take care of yourself. You can do it. Remember that. Bye-bye.